morning, students. Today let us know about Unit 5, which includes phylum Mollusca, phylum Echinodermata, phylum Hemichorta. Firstly, let us know about phylum Mollusca. General characters of phylum Mollusca. They are mostly found in marine and freshwater. Very few are terrestrial and found in moist soil. They exhibit organ system level of organization. The body is divided into head, visceral mass, muscular foot and mantle. The head comprises of tentacles and compound eyes. The body is covered by a calcareous shell. The muscular foot helps in locomotion. They have a well-developed digestive system. The radula is the rasping organ for feeding. Respiration in mollusca occurs through the general body surface, gills or pulmonary sac. The blood circulates through the open circulatory system. They have a pair of metanephridia that helps in excretion. The nervous system in mollusca consists of number of paired ganglia and nerves. The tentacles, eyes, ostracium and statocyst act as the sensory organs. The sexes are separate in most of the molluscs, but some species are hermaphrodites. Fertilization may be external or internal. Next, classification of phylum Mollusca. Phylum Mollusca contains the following subgroups. Class Eplacophora, and it consists of solenogaster, deep sea worm-like creatures. In next class, bivalvia, clams, oyster, scallops, mussels. And in next class, cephalopoda, squid, nautilus, cuttlefish will be present. And next class, gastropoda. In gastropoda we can see snails, slugs, limpets, sea hares. In next, monoplacophora, deep sea limpet-like creatures are present. Next class, polyplacophora, they are chitons. Next, lastly, scaphopoda, these are tusk shells. Next, let us know about important question, pearl formation in pelecypoda. The pearl of oyster produce pearls as an adaptation against outside material. When the foreign material enters the body, such as a particle of sand or a small parasite, it adheres with the mantle. The mantle epithelium starts secreting concentric layers of nacre around the foreign material. The completed structure is called pearl. Most precious oysters are found in pearl oysters of the genus Pinctada. In diagram we can observe this one is the foreign material. It adheres into the mantle. And this layer is called the mother of pearl, and this layer is called as mantle, the inner part. This is the foreign body. It adheres with the mantle. And here layers of nacre will be found. Next, inside pearl formation is happened. Next, culture of pearls. The culture of pearls is a complex but sensitive process. It involves the following steps. First one, collection of oysters. Second one, preparation of graft tissue. Third one, preparation of nucleus. Fourth one, implantation. Fifth one, rearing of oysters. And last step, harvesting. Next, collection of oysters. Oysters for pearl culture are obtained by three methods. They are as follows. Pearl oysters are collected from the bottom of the sea. Spats, which are also known as young oysters, are collected by placing cages in spat falling areas of the sea. Next, in the laboratory, eggs of pearl oyster are fertilized and egg ones are obtained. Next, next topic, torsion in gastropoda. Torsion is the process by which mantle cavity and visceral mass are rotated 180 degrees in a counterclockwise direction. Let us know about mechanism of torsion. The only known animal that can rotate completely or 180 degrees using just mantle contraction is Echinodermata. In a 90 degrees rotation was achieved in two stages. The first by the contraction of the larval retractor muscles and the second via slower growth. It is the most prevalent mechanism currently understood. It is seen in the patella and haloides. Rotation 180 degrees solely via differential growth mechanism such as in vivipara. Rotation caused by distinct growth stages whereby the anus attains an adult stage appropriate position such as in aplasia. Torsion is no longer recognized as a movement of viscera pallium as organs have been in the post-torsional posture since their initial development. It is observed in Adalaria. Next, stages of torsion. Torsion is achieved in two stages. In stage 1, it takes into account the 90 degrees rotation caused by muscular contraction and the shifting position of the mantle from the posterior to the anterior end. Next, in stage 2, it takes into account the remaining 90 degrees and involves differential growth. Next, effects of torsion. The following are the some of the events that occur during torsion. First one, looping of the digestive or alimentary canal. It was originally straight from the mouth to the anus and is now thrown into a loop after torsion. Second one, displacement of the mantle cavity. It is primitively posterior and is now shifted forwards. Next, third one, loss of symmetry. Due to torsion, the bilateral symmetry of molars is lost. Next, fourth one, chiastoneuria. The pleurovisceral connectives become twisted. Next, last one, changes in position. Before torsion, tenidia, renal orifices and anus point backwards and the auricle is positioned behind the ventricle. After the twist, torsion, tenidia, renal orifices and anal project forward and the auricle lies front of the ventricle. Next, phylum Echinodermata. Characteristics of Echinodermata. They have a star-like appearance and are spherical or elongated. They are exclusively marine animals. The organisms are spiny skinned. They are triploblastic and have a coelomic cavity. The skeleton is made up of calcium carbonate. They have an open circulatory system. They respire through gills or cloacal respiratory tree. They have a simple radial nervous system and the excretory system are absent. The body is unsegmented with no distinct head. The mouth is present on the ventral side while the anus is on the dorsal side. Next, the tube feet aids in locomotion. Fertilization is external and the development is indirect. They possess the power of regeneration. They have poorly developed sense organs. These include chemoreceptors, tactile organs, terminal tentacles, etc. Next, this is the classification of phylum Echinodermata. Here we can observe Echinodermata is undergoes into three subphylums: Crinozoa, Asterozoa and Echinozoa. In each subphylum, in Crinozoa, it belongs to the class Crinoidae. In this Crinoidae class, we can observe sea lilies and sea feathers. In Astrozoa, it again divided into two classes, Ophiuridae and Astoridae. Next, Echinozoa. Echinozoa undergoes into two classes. Echinoidae. In Echinoidae, we can observe sea urchins and hurt urchins. Next, Holothuridae, we can observe sea cucumbers. Here, brittle stars and starfishes. This is the classification of phylum Echinodermata. Next, water vascular system of starfish. The water vascular system is a modified part of coelom. It consists of a system of seawater filled canals having certain corpuscles, and it is otherwise called as umbilical system. Their function are locomotion, food capturing, and third one, respiration. The essential parts of water vascular system are madreporide. In the middle of the body, we can observe the madreporide. Next, stone canal. Next, ring canal. Next, radial canal, Tideman's body, Polian vesicles, lateral canals, and tube feet. We can observe all these essential parts in water vascular system. These are the main parts which involves in water vascular system of Echinodermata. Next one, larval forms of Echinodermata. In echinoderms, eggs and sperms are released in water and fertilization takes place in water forming zygote. Echinoderms are deuterostomes and hence cleavage is radial, holoblastic is holoblastic and indeterminate. The larval hatch in water and feed and grow through successive larval stages to become adults. And first larva, Bipinnaria larva. It is the larva formed seen in the class of Asteridae. The fertilized egg is homolecital. It undergoes holoblastic cleavage and develops into blastula and gastrula stages. The gastrula elongates in length and gives rise to Bipinnaria larva. Below we can observe the diagrammatic representation of Bipinnaria larva. Next, Brachyolaria larva. Bipinnaria larva swims for few weeks in the seawater. It finally transforms into next larval stage called Brachyolaria larva. This is the diagrammatic representation of Brachyolaria larva. Next, Auricularia larva. In Holothuridae, this larval form is seen. It is the free swimming pelagic larva. Arms are absent. Elementary canal is developed. It opens with mouth and ends with anus. Intestine is curved. This is the diagrammatic representation of Auricularia larva. Next, Ophiopletus larva. This larva is seen in the life history of Ophiuridae, like brittle star. Next, it shows many long arms. It is bilaterally symmetrical. Below we can observe diagrammatic representation of Ophiopletus. Next, Echinopletus. It is seen in the life history of Echinoidae. It is a microscopic larva. It swims in water. This larva shows ciliated bands which are developed into arms. Next, Doliolaria. In crinoidae groups of animals, the larval form is Doliolaria larva. It is a free swimming larval form. It contains an apical tuft of cilia which will be sensory. Here we can observe. Here at the top of the body larva, apical tuft of cilia which will be sensory in function. This is the diagrammatic representation of Doliolaria. Next, general characters of Hemichorta. The body of Hemichordates is worm-like, thus they are commonly called worm animals. They have a bilaterally symmetrical body and are triploblastic in nature. They have a true coelom. Proboscis, collar and trunk are the three main parts of the body. Gill slits are present that perforate the pharynx. From the roof of the buccal cavity, a buccal diverticulum arises. The buccal diverticulum is also known as the stomochord. It is a projection of the digestive tract. Examples of Hemichorta include balanoglossus, sacroglossus, etc. Here we can observe the phylum Hemichorta structure. Here the proboscis, here mouth and coelom is present, and here these lined dot-like structures are called as gill pores. The middle part is called as trunk. Next, balanoglossus structures and affinities. The body of balanoglossus is soft, elongated, cylindrical, bilateral, symmetrical, being richly ciliated all over and covered with mucus. The length of animal varies from 2 centimeters to 2.5 meters according to species. Most forms are dark colored, though reddish tints are present. Several species are luminescent due to mucus. They have an offensive odor. This is the structure of balanoglossus. The top of the part is called as proboscis and this is called as collar. Internally we can observe the branchial region, and here we can observe genital wing. This middle layer is called as middle dorsal ridge. This is called as hepatic CK. This one is hepatic region. The last portion of the part is called as anus.